And Dr. Saykin is the director of the Indiana Alzheimer Disease Center, as well as director for Center for Neuroimaging. In short, he made a significant contributions in our understanding of cognition and memory and associated disorders using multiple approaches that he's going to talk about. Dr. Saykin is an original thinker, erudite scholar, and prolific author. He has over 600 publications with an impressive H-index 106. In addition, Dr. Saykin is the PI and co-investigators of numerous NIH-funded grants, over 50. So today he's going to talk about the IADRC as research hub on the path to precision medicine. So without further ado, please join me in welcoming Dr. Andy Saykin virtually live. All is yours, Andy.
Thank you, Deb. Well, I'd really like to welcome everyone to our scientific symposium this year. We have a really exciting lineup of speakers and a great theme, thanks to Deb Lahiri, who's organized this. You've just seen all of the talks, I think, scrolling, and I won't go over them again. But I do want to just take another moment to thank Deb Lahiri, who has been organizing these every year. We have a great theme in terms of biomarkers and a broad range of related topics. And also to really sincerely thank Brian Maloney, Donna Wirt, Sarah Van Hayden, Joan Mason, Andrew Tackett, Alan Beard, and others who have really pitched in to help make this all work today. And I especially want to thank our wonderful speakers, both our national level speakers visiting virtually this time, and also our local speakers from the center. These are my disclosures. Our center was just renewed for another 5 years after 30 very successful years contributing to Alzheimer's disease and related disorders research. I always like to begin by just reminding all of us about the key themes. I mean, our overarching theme is really developing the knowledge base for precision medicine of Alzheimer's disease and related disorders, and fostering innovative research at all levels, from etiology to detection and treatment and prevention, developing resources and infrastructure, helping to identify and prioritize targets, to increase the range of investigators with expertise in Alzheimer's disease research from multiple disciplines, including a new emphasis on data science and translation, and also to provide educational and training opportunities for both professionals and also members of the community as providers and as informed citizens involved in supporting research going forward. So the theme that I increasingly like to talk about is the growing hub and really the ecosystem of Alzheimer's disease and related neuroscience and neurodegeneration research here at Indiana University and beyond, with connectivity throughout the US and also abroad. And just to, you see the matrix of, of icons of all the related programs that IU is either leading or co-leading or affiliated with. But, just to call attention to several new developments, there are now 2 T32 training programs. One that was funded over the last year, led by Gary Landreth, that is neuroscience oriented. We have our first cadre of pre and postdoctoral trainees enrolled in that. And then a very new T32 was just developed for MD PhD candidates that focuses on social sciences. So that's fantastic. There are several new cryo-EM related projects led by Drs. Vidal and Getty and collaborators at Purdue University and beyond. There are some new COVID-19 studies led by Sophia Wang, who's also our ORCOR lead. And these are in collaboration with NAC and with other universities, and then an R21 locally here. Very important emergent topic, obviously, that's impacted the last year and a half tremendously for all of us. A brand new R24, a collaborative for aging research and engagement led by Yvonne Liu, with several very prominent, prestigious MPIs as well. That's a really important direction now in terms of health disparities and just increasing the diversity of the research base and methods for doing that. A couple of other new programs I want to mention in addition to the major consortia already listed here, at least with their icons. One is a new grant over the last year applying artificial intelligence strategies to Alzheimer's disease, especially targeting the genome in Alzheimer's disease and whole genome sequencing data, but also the complex phenotypes from neuroimaging and other biomarkers and cognition from across studies. This is a spin-off from the Alzheimer's disease sequencing project that we're very excited about. A few more words on that later. And then another, the K-base grant, which is a new UO1, bringing the Asian population in Seoul, Korea, and it's an ADNI-like grant that I'll say a few more words about later. And then, the whole other landscape of ADNI leads, Diane, and so forth, NICRAD, and then the U54s, Model AD and Treat AD for drug development and model systems. So it's a very exciting time here, and the great challenge for all of us is to obtain the maximal synergy from all of these different studies and resources, and to not only engage in new discovery, but also to use this as a vehicle for training the next generation of researchers. So a major theme today is biomarkers, but biomarkers are really in service of something, and I would argue that they're in service of precision medicine, trying to really understand on an individual basis what the pathophysiological profile is and risk factors early on, and then to be able to tailor the treatment for that individual, the proverbial, right treatment for the right patient at the right time. And a major transformation in the field has really been the development of the framework of using biomarkers to develop a biological definition of disease, and then to target interventions accordingly. So there are many types of biomarkers to set the stage for today. One is a predisposition or risk biomarker. So if you think of ApoE as the strongest genetic risk factor for sporadic Alzheimer's disease, that's identifying individuals at risk. Diagnostic biomarkers, on the other hand, like amyloid or tau PET, CSF assays or blood assays, are identifying the current state and are useful for detection and diagnosis. So these are really complementary. Then, of course, there are also prognostic and predictive biomarkers that play a role in understanding and predicting disease trajectory and who may respond in what way to what particular treatments. And these are becoming increasingly important as well. So while we talk a lot about amyloid and tau, whether it's PET or CSF or now blood, it's really important to remember that it's the brain as an organ system has many pathways and also lives in connectivity with a much broader environment, everywhere from multiple organ systems through to ultimately even the sociome and environment. And we have to keep a broad perspective while we also zero in at a molecular level, and even down to the level of the cryo-EM studies that Drs. Vidal and Getty are doing to understand the nuclear basis of abnormal proteins. All of this is in a context, and probably all will be very important ultimately for effective treatment and prevention. There are many pathways beyond amyloid and tau that probably are contributing to the development, deposition, and clearance or failure to clear these abnormal misfolded proteins. And it's important, I won't go through the list that you can see here as icons, but all of these play a role, and we need to understand for a given individual what role these different factors play. You just to pick randomly blood-brain barrier, neurogenesis, and turnover, phagocytosis, endocytosis, and abnormal protein processing, and beyond. So we need to keep a broad perspective, think of all of these pathways, and then try to prioritize those pathways for a given individual and for a given treatment strategy. Genetics has made remarkable progress, and in part this is due to the rapid accumulation of very large-scale data. So superimposed on the proverbial Jack curves, or the ADNI icons for amyloid, tau, neurodegeneration, and cognition, we really can think about the role of genetics, both individual genes and especially polygenic combinations of genes, in terms of their impact on these different biomarkers or different components of disease that are stage specific. So we need to be able to identify risk genes, but also protective genes. And all of these genetic signals may provide clues to novel targets that can be used as a basis for intervention. And the database that's enabling interrogation of the role of the genome is really growing. So, just last month, a large-scale meta-analytic GWAS study was published, and it's now up beyond 1.1 million individuals. So this is really extraordinary. We're finally getting to the point of having the statistical power to really identify the role of genetic variation. And going from GWAS array data, which is still very powerful, but going all the way to whole genome sequencing with very large numbers is going to really enable the interrogation of some of the less frequent variants and to understand their role in the various pathways that were on that prior slide. So what do we do with the small genetic signals, though, as we have statistical power with over 1 million individuals to detect signals? They're small signals, and they have to be aggregated to be really meaningful. So a very prominent role now is the polygenic combination. And there are many studies on this. There are polygenic arrays and tools that are headed toward clinical application. But we have a lot to learn, especially because most of the data collected so far are people of European ancestry, and there's a tremendous need for new data on other racial and ethnic groups to fully understand the range of biology, and so that we can ultimately understand how the basic genetic architecture intersects with the environment, social and environmental factors to lead to risk, and also for development of therapeutic strategies that target those affected pathways. So I think the future is not only an overall polygenic risk score that adds up small risk, but scores that are targeted toward amyloid, toward tau, synaptic loss, vascular changes, immune changes, and other pathways. And those scores in combination may help tailor treatments to the individual. In addition to looking at DNA variation in these scores, I think the next wave in functional genomics is really to understand the role of the transcriptional factors, so that we see genes that are upregulated, downregulated, or otherwise altered, and epigenetic changes like DNA methylation or other epigenetic marks that may play a role. Metabolomics and lipidomics also are likely to be able to be composed as either polygenic or polymodal scores that are helpful for tailoring precision medicine in the future. I think you'll hear more about this from some of our speakers going forward. phenotypes are expanding as well. In some nice work recently published by Mei-Chin Yu, a postdoc in our group, it's basically mapping between the connectome, Alzheimer's disease pathology, and gene expression. And I think the combination of integrating these complex data sets as more and more data becomes publicly available to the community is going to enable a deeper understanding of the intersection of pathology with brain function, which is ultimately what we want to preserve. Some of these new consortia that I mentioned are very exciting. The AI for AD spans multiple institutions around the country, led by Paul Thompson as our contact PI, and then 5 of us in all are the multiple PIs at Penn and at Pittsburgh, as well as Indiana. But all of this is taking that wonderful information available from so many consortia, from the Alzheimer's disease sequencing project, from AMP-AD, the Accelerating Medications Partnership Program, and much more, and figuring out how to integrate that information using powerful analytic tools. So a large-scale national team effort is underway for this that we're quite excited about. The other development I mentioned, that's a collaboration spanning continents with Seoul National University, but then also University of Pennsylvania, Miami for genome sequencing, Lonnie at USC for storing all the scans and so forth, is the K-base program. So this is an ADNI-like cohort with amyloid and tau PET, cognitive studies, longitudinal follow-up, multiple omics on a Korean Asian population that will also be widely shared and available for analysis, and represents a new and exciting collaboration co-led by Dongyoung Lee at Seoul National University and Kwansik No here at IU. All of this data requires great analytic resources, and we're excited that Indiana University's latest supercomputer, Big Red 200, is about to go live and enable us to send all those genomes with the 3 billion base pairs and all of the voxels and omics data to Big Red 200 for computational analysis, as well as all the work in the cloud with the other collaborators and universities. So all of this converges for me basically here. We have biomarkers, today's theme, driving precision medicine, where for each individual, we want a unique individualized characterization of their profile, and where the multiple omics data, proteomics and other assays, genetic risk, lifestyle factors. At the end of the day, you'll hear about digital biomarkers from Rho to O. You know, all of this information has to converge. We need a database in a diverse populations to really understand these mechanisms and tailor interventions. And ultimately, in this, cartoon-like description, you have amyloid, tau, immune dysregulation, and then environmental factors, diet, metabolism, and the microbiome, and so on, lifestyle factors. All of that needs to be tailored into a strategy for the individual. People differ, their genetic backgrounds differ, their environments differ, their lifestyles differ, and we ultimately need a customized, tailored intervention for the right one for the right person at the right time. And things are very promising now because so much data is becoming available, but we have to figure out what data is missing and obtain it quickly. So that's just a very 30,000-foot overview of some recent developments and the convergence of resources and programs here at IU. I want to thank all of the collaborators from all of the different entities that were on the earlier slides, and my colleagues here locally and then nationally and globally working on these different programs. It's an exciting time.